morning, students. This is description on the structure of nephron as a part of the general physiology topic. So let's go to the screen sharing. Okay, so structure of nephron. Nephron is a part of the functional unit of the kidney. They cleanse the blood of toxins and balance the constituents. Homeostatic set points in the process of filtration, reabsorption, and secretion. The nephrons also function to control blood pressure by production of the brain and red blood cell production by homocysteine and calcium absorption via the conversion of calcium into calcium via the active form of vitamin D. Each nephron consists of a blood supply and a specialized network of ducts called a tubule. And for each nephron, an apparent arteriole feeds the high-pressure capillary that called the glomerulus, and blood is filtered by the glomerulus to produce a fluid which is caught by the nephron tubule. Now it will be called the filtrate. The proximal tubule that surrounds the glomerulus and catches the tubular fluid is called the glomerular Bowman's capsule. You can refer to the picture that is coming over in the right side of the slides. The glomerulus and glomerular capsule together form the renal corpuscle, and the filtered fluid poured by the glomerular capsule travels to the rest of the tubule to the proximal convoluted tubule, loopal tubule, and distal convoluted tubule, and in this order, before exiting the nephron into something like a collecting duct shared by many nephrons. All the nephrons, the glomerulus and the cortex, some nephrons have short breaks of energy that do not dig far beyond the cortex. These nephrons are called the cortical nephrons. And about 50% of the nephrons have very long breaks of energy that extend deep into the medulla and are called the juxtamedullary nephrons. And blood flowing nephrons like the eyes, particularly by the tubular capillary, which is retains the substances from the tubule. The efferent arteriole is the connecting vessel between the glomerulus and the peritubular capillaries as vasa erecta. The blood exits the glomerulus into the efferent arteriole. The efferent arteriole informs a second capillary network around the tubule called the peritubular capillaries. The juxtamedullary nephrons, the portion of the capillary that follows a loop of medulla deep into the medulla is called the vasa erecta. The glomerular fluid progresses through the tubule. These capillary networks recover most of the solutes and water and return them to the circulation. Capillary bed drains into a vessel that forms a secondary capillary bed. This is another example of something known as the four-cone system. When the microanatomy of nephron exists, the first one is the renal corpuscle. The renal corpuscle consists of glomerulus and the glomerular capsule. The glomerulus is a high pressure fenestrated capillary with large holes for fenestrations between the endothelial cells. The glomerular capsule captures the filtrate created by the glomerulus and directs this filtrate to the proximal convoluted tubule, which will be commonly known as PCT. The ultimate part of glomerular capsule is a single squamous epithelium, which I see this is in the squamous epithelium picture also shown over here. The transitions over the glomerulus as uniquely shaped cells with finger-like arms. They are also pedicels that cover the glomerular capillaries, and a thin basement membrane lies between the glomerular endothelium and the sporocytes. The pedicels interdigitate to form something like a filtration sleeves, leaving small gaps that form a seal. The blood passes through the glomerulus, almost 20% of the plasma filters out through these fenestrations through the basement membrane, and between these seal-like fingers there is a podocyte to be captured by the glomerular capsule and funnel to a proximal convoluted tube. And these features comprises about something known as the filtration membrane. Now, you can see the podocytes. The podocytes integrate with the structures called cells, okay, and filter substances into the glomerular capsule. In the figure A, we can see the large cell body can be seen at the top right corner. This is a cell body which branches extending into the cell body. From the cell body, and the smallest finger-like extensions are the baby cells. Okay, and you can see situations where they have these are the cell bodies, and this is a capillary tube which this is the fenestrated capillary, and the fenestrations means this holes here you can see over here, and these fenestrations allow many substances to leave the blood base primarily on their sides. The filtration membrane prevents the passage of blood cells or large proteins and most negatively charged particles, but allows most of the other constituents to. These substances cross readily if they are less than 4 nanometer in size, and most pass freely up to 8 nanometer in size. Negatively charged particles have difficulty leaving the blood because the proteins associated with the filtration membrane are also negatively charged, which will repel them and only allow the positively charged substances to pass more readily. But there are also mesangial cells in the filtration membrane that can contract to help regulate the rate of filtration of the glomerulus, and this results in the formation of the filtrate that does not contain cells or large proteins and has a slight predominance of positively charged substances. Then comes the proximal convoluted filtrate. The fluid collected by the woman's capsule lingers in the proximal convoluted tubule, and the simple cuboidal cells, you can see simple cuboidal cells that you have the structure over here. Simple cuboidal cells form this tubule with a prominent microvilli on the luminal surface forming a brush border. These microvilli create a large surface area to maximize the absorption and secretion of solutes in the filtrate, the most essential function of this portion of the vessel. These their membrane, and so they possess a high concentration of mitochondria in order to produce ATP, because all these ions eventually they are transported across with the help of ATP. That they are active transfer. Next is a loop of Henle. This is the loop of Henle. The descending and ascending portions of the loop of Henle are continuous at the same tubule. They run adjacent and parallel to each other after having made a hairpin turn at the deepest point of the descent. The descending loop of Henle consists of an initial short, thick portion, and long and thin portion, whereas the ascending loop of Henle consists of an initial short, thin portion followed by a long, thick portion. And the descending thick portion consists of a simple cuboidal epithelial cell similar to the proximal convoluted tubule, and the descending and ascending thin portions consist of simple squamous epithelium. And different portions of the loop have different permeabilities for solutes and water, and the ascending thick portion consists of simple cuboidal epithelium similar to that of the distal convoluted tube. Now let's say distal convoluted tube. This is a distal convoluted tube. Okay. Like the proximal convoluted tube, it is formed by simple cuboidal epithelium, but is shorter than the proximal convoluted tube. You can see how short it is. These cells are not as active as those in the proximal convoluted tubule, and there are fewer microvilli on the epithelial surface. However, these cells must also pump ions against the concentration gradient. The large numbers of mitochondria, also fewer than in the proximal convoluted tubule, are present. Collecting ducts. The collecting ducts are continuous with the nephron, but not technically part of it. Duct several nephrons for final modification. Collection ducts merge as they descend deeper in the medulla to form the terminal ducts which empty at the back. Then to just a glomerular because they are lying just outside the Bowman capsule and the glomerulus. And that's, they are like they have the afferent and efferent arterioles enter and leave the Bowman capsule. We have seen the wall of the distal convoluted tissue at the point forms a part of the glomerular and adductus. This cluster of cuboidal epithelial cells monitors the fluid composition of fluid flows passing to the distal convoluted tubule. And the second function of the enzyme is to regulate the renin release from the glomerular cells of the afferent. This you can see the glomerular apparatus and the thank you very much.